Alright, I'm going to switch to the iPad to try to draw this for you, so the sound quality will change slightly, I'm sure. Okay, so the first step of mitosis is prophase. So we have our nuclear envelope and our plasma membrane. Let me draw the nuclear envelope inside here. And we're going to draw a cell today that is 2n equals 4. So a diploid number of 4. So there's 4 total chromosomes. I'll draw 2 of them as red. We'll put one there. They're just kind of randomly arranged and they're condensing here. And I'm going to draw 2 of them as blue. The other thing that's happening here is our spindle is forming. I'll draw it here. Here's the 2 centrosomes close together. There's the spindle forming between them. Okay, that's prophase. Number 2, metaphase. Okay, so we're going to draw the plasma membrane. We're going to draw the nuclear envelope breaking down here. We are going to draw our chromosomes. As before, and the spindle. The spindles have moved to the opposite ends, and we are now attaching microtubules to these chromosomes. So these are kinetochore microtubules. And then out here are non-kinetochore microtubules. Now, if we were going to zoom in on one of these, because it's kind of a mess here, we would see the here's the chromosome. And we have one microtubule attaching to this sister chromatid and one attaching to this sister chromatid. Okay. Now we move to, so prometaphase, now we move to metaphase. And in metaphase, it's pretty easy to identify. Here's the cell. And we're going to have, oh, that's the wrong button. We're going to have the chromosomes lined up at the metaphase plate, which is like an imaginary, let me keep that, it's like an imaginary equator here, okay? There they are. Here's the microtubules. And they're all lined up in the middle, okay? That's metaphase. Now, you should be able to draw along with this and make your own version of this. Maybe yours will be a little cleaner than mine, but that'll help you to understand what's going on here. Now, number 4, Anaphase. And finally, number 5 down here will be Telophase. Okay. In Anaphase, our cell's elongating a little bit there. Our sister chromatids, I could go back and see how I drew these. I guess I'll put one there, one here, one here, one here, and then blue, one there, one here, one here. Okay, so what's happening here? Sister chromatids have pulled apart and they've become daughter chromosomes, and they're being pulled in opposite directions. So that one and that one are now daughter chromosomes. In telophase, We have a cleavage furrow forming here. That's when the cytoplasm is pinching in half. We have the formation of new nuclear envelopes. The spindle's going away, or has gone away. Our chromosome, actually I'm going to switch to this highlighter. Let me make it smaller though. The chromatin is, the chromosomes are decondensing and forming chromatin. So I'm gonna draw it as kind of a cloud in here rather than as distinct chromosomes. So once this is all done, we're going to have 2 new cells produced. And those 2 new cells, if we are able to see their chromosomes condensed, would look just like the cell that we started with in prophase, essentially, except, let me go back to this slide here. I want to show you the cell that we really started with. Let me draw that initial cell up here in the corner. That initial cell, before it went into S phase and duplicated its chromosomes, if we were able to see its chromosomes, which we could not because they'd be decondensed, but if we could see them, they were wound up tight, we would see in a cell of 2n of 4, we would see I'm just going to draw them in here kind of like that. 4 chromosomes, each consisting of 1 chromatid each, right? And at the end of this, if we were able to condense this chromatin here in these 2 cells to see what it looked like, we would have the same situation. We have basically produced the same cell that we started with. Now, when these cells get ready to divide, what will happen? They will copy their DNA, so their chromosomes are going to look like X's again. Once they copy their chromosomes, right? There'll be 4 of those little X's in there, and they will go through the same process when they divide. But these 2 new cells that have just been produced are now in G1 phase. They're going to grow, get bigger, and prepare to divide. So those are the stages of mitosis, and hopefully you are capable of drawing them yourself, given a number like 2n of 4 or 2n of 6. Whenever you're doing mitosis and you see 2n of 4, that means there's going to be 4 X's that you draw. 2n of 6, there's going to be 6 X's that you draw. That's how you can tell. And when I talk about that, when I say something is 2n, I might ask you, what is the ploidy? By ploidy, I mean, is it diploid or haploid, right? So if the ploidy is diploid, it's diploid, and it's 2n of 4, that would mean there's 4 chromosomes in there. If it's 2n of 46, like a human, that'd be 46 chromosomes in there. So we'll use that terminology as we go. If you don't quite get it yet, I'll introduce it again in a bit.